All right. Thank you all for being here and welcome to Speaking of Oats. Speaking of Oats is a partnership between Oat Global and the Oat newsletter. I'm Jim Bradeen, professor at the University of Minnesota and director of Oat Global. And we're joined today by Charlene White, who is, of course, the curator of the Oat newsletter. And we're very happy that you're here to talk about oats today. Before we get started, just a few quick housekeeping comments. First of all, this is being recorded, so if you miss any part of this, it will be posted to YouTube. You can find the recording by searching for Speaking of Oats. You can access it through the Oat newsletter page or through the Oat Global page as well. And at the bottom of your screen, you will see both a Q&A widget and a chat widget. We'd like you to use the Q&A widget for any questions that you have for our panelists. That helps us save those questions as part of the recording for later. Meanwhile, if you've got general comments, feel free to use that chat function. Charlene, before I introduce our speakers today, if I missed anything.
No, I think you pretty much covered it. Thanks, Jim. Hello, everybody.
Great. So it's our pleasure to welcome three scientists today who will be speaking about advancements in oat quality. We're joined by three scientists, research scientists from Agriculture and Agri-Food Canada. First, we'll hear from Dr. Nancy Ames. Second, we'll hear from Dr. Lovemore Malunga. And finally, we'll hear from Dr. Sijo Joseph. And I'm going to turn it over to Nancy for the first portion of this session. Thank you for being here, Nancy.
Great, thanks so much, Jim. And thanks, Jim and Charlene, for inviting us. I'm going to start this off. I'm really going to introduce our work. The three of us all work at the same location, the Richardson Centre for Functional Foods with Agriculture and Agri-Food Canada, and we all work on oat quality. So the title of this today is Advancements in Oat Quality, really focused in on the idea that, some of our research work we think is looking at new ways to define quality, new quality criteria, really looking at oat improvement and possibly new uses of oats. So this is kind of all encompassing the work we do that we're calling advancements in oat quality. Okay, so now it doesn't move along. Okay, so when we think about advancing oat quality or looking at possibly, new ways to look at oat quality, I think it's really important for us in people that work on oat quality or work on oats and end products to continually evolve, where we're going with oat quality, what are we measuring, what are our criteria? And one of the reasons for this is kind of obvious from this slide is that there's so many new products, there's so many new things out there that could have different quality criteria, could benefit from looking at oat quality in a different way. There's clearly a lot of interest in new oat products. See here, like we've said, new, not old-fashioned oats, but of course, we're also interested in old-fashioned oats. So all of these things, all of these different products may mean that we have to look at different quality criteria or possibly looking at new ways to, evaluate oat varieties for fitting into these different products. So are there different varieties that suit being made into, oat spaghetti versus oatmeal or overnight oats? Are they the same as some of the other products? And of course, the other thing to think about is consumer perception and market trends. Here's just an example of like the media frenzy that seems to be around oat milk. And so whether this is a trend or not, really, it's a really popular product. Everybody knows that that's on this call today. But really, the industry is just catching up. And by the industry, I mean, all the people that work on oat quality and all the way along the value chain, we're really just catching up on what raw material has the best characteristics, for oat milk, for the processing of it. What are the best techno-functional properties of oat protein? These are also important. There's a lot of interest by consumers looking at plant-based proteins, and also plant-based proteins being used in oat milk, oat proteins. And, what are the techno-functional properties of those oat proteins? This is something that we don't really look at on a regular basis when we're thinking about oat quality. So these are some of the advancements that we have to really be moving forward on to be able to keep up with consumer trends. So here is, in Western Canada, the list here is the oat quality targets that we look at if a variety or an oat variety is going to be registered. So this is, one example of traditional oat quality evaluation. But at the same time, these targets, the target values themselves, are continually changing. The oat industry gets together and really updates these breeding targets. And so they don't stay the same. In fact, this may not be even as up to date as it currently is. This slide is a couple of years old. So it could be that some of these targets have changed because the committees that oversee oat quality for the registration of oat varieties might have changed some of these targets. So that's kind of part of the evolution and keeping up with advancements in oat quality. And that's just a look at what the current targets are. When we do talk about oat quality, we really want to think about who we're talking to in the value chain, because depending on who you are in the value chain, your quality criteria or your quality requirements or your interest in particular quality areas might be quite different. And quality research needs to really consider the varying needs along the value chain. For example, there are processors that have specific or have quality specifications that might not be one of those target criterias that I just mentioned. Possibly something like RVA pasting limits and that sort of thing. There's a number of characteristics that aren't, really something that we traditionally look at when we're evaluating new breeding lines or our breeders look at. But when you look across this value chain, all of the people along this value chain are interested in oat quality. They all have a stake in some aspect of oat quality, and they have their own definition. And in many cases, they're the same thing, but they're calling it something different. But one of the areas that's maybe least considered is maybe this consumer column here. Possibly some of the consumer quality aspects are not always considered by everybody along the value chain. So this is one of the areas that we focus in on in our research, while always trying to work with the other members of the value chain. But some of the things I'm going to talk today about, and then my colleagues really relate to that consumer part. And and specifically in the consumer part, the health benefits and the food safety and even end products. So that's some of the areas we've been looking at. And some of our advancing research or research on advancing oat quality is taking a look at Enanthramides, and we're all aware of what Enanthramides are. And my colleagues, Sijo Joseph and Lovemore Malunga, will be going into more details on this. I'm just really introducing that some of the areas that are of interest to all of us really with respect to Enanthramides might be composition and some of the health benefits. Beta-glucan, looking at benefits beyond cholesterol lowering. So we know that we've got health claims on beta-glucan for cholesterol lowering, but there's also other potential health benefits that we're looking at. And Some of these depend on the physiochemical properties, which are something we don't typically measure when we're thinking about quality criteria. We normally look at beta-glucan content, but beta-glucan viscosity, molecular weight, concentration, and all contribute to, and solubility, all contribute to viscosity. And so this is something that, many places are looking at this more and more, but it's not typically looked at when we're evaluating breeding lines. So this is something that we know is, very soon in the pipeline to looking at this particular quality criteria. The other thing that we're looking at a lot is looking at functionality of macronutrients. This comes into protein, looking at protein and starch, these two big nutrients that are in oats. Protein and plant-based proteins, as I mentioned previously, are becoming quite important to consumers. People are really interested in protein extractability, but these are some of the things that we're measuring. Different varieties may respond may be more extractable than others in terms of protein, the digestibility of that protein, what's left over, the starch digestibility, also end product testing. These are something that we don't typically measure, but could play a big role in the future, and maybe something to be thinking about as we're going ahead with advancing quality criteria. Just a quick look at how we look at genotype, environment, and processing with respect to all these quality traits. So this is just a list. I'm not going to go, it's some of the things we're working on, just to consider that when we're thinking about Enanthromide composition, we know that genotype, environment, and processing affects the composition, and also which particular Enanthromides are there, and what quantity and what ratios. We know that these G by E by P affect beta-glucan viscosity. We need to know whether G by E by P affects the amount of amount of beta-glucan that might affect blood glucose or blood pressure. We need to look at G by E by P in respect to protein digestibility, and all of the other factors that we're looking at. We have to consider it from this perspective. This is just a quick look at why it's important. Here's even flake quality. We don't look at flake quality a lot traditionally, but this is just a study where we looked at the environment, different ways to harvest, and the result was different flake quality depending on how something was harvested. Now, I'm not going to spend any time talking about our experimental capabilities, but it's here for later if you want to look at it and see what kind of work we do. But it is my introduction to move on. Also, just a quick look at our oat processing facilities at the Richardson Center. But this is my introduction to move on to my colleague, Dr. Lovemore Malunga, who's going to talk a little bit more about some of the detailed work that we're doing that I've just introduced to you. So I'd like to now introduce Dr. Lovemore Malunga to take over here.
Thank you, Nancy. Okay. Okay, I hope everyone can see the slides.
Lovemore, we're not seeing it in presentation mode. Could you swap screens?
Oh, okay.
That's perfect. Thank you.
Okay, thank you. Yeah, so thank you everyone for joining. My presentation now will focus mostly on giving you some insights into what we actually work on in our program. But by now you have seen that from what Nancy has already mentioned, that we run a very comprehensive oat quality program. And we try to follow the changes at the different point or steps in the processing. For example, raw versus kilned, or whether tempering or microwaving, or whatever processing that may be applied to oats, what effects or changes they would make toward the functional and the nutritional quality of oats. And I just also wanted to mention that most of our methods, we try to follow official methods of analysis, and wherever we don't have, we try to develop our own methods, and hopefully one day they could be adapted for as official methods. So I'll try to go over what we do, but in the interest of time, I'll just try to introduce, we'll give a brief report on what we are actually doing, and hopefully that will trigger some discussion points later where we can actually go into details. Or if you are more interested, we have our contacts at the beginning of the slide. So feel free to contact us and we can discuss further into that. So first of all, I'll go into protein research, as we are all familiar that at the meantime, we are riding the protein wave. And thankfully, in our program, we are also trying to support the development of innovative processes for obtaining high protein ingredients. So in this particular case, we try to look at developing different extraction or concentration methods. And we try to identify which of the Canadian oat varieties are suitable for protein. And we also know that protein itself is not good enough, but we need to understand its properties so that how can it be used in the industry, as well as even if it's usable in the industry, modern times we need to substantiate with research on why oat protein might be good for our for us or for our health. So in our labs, we try to focus on looking at when we look at the oats, we try to look at different oats, as we are all familiar that oat protein content varies greatly, as well as the oil. And we try to look at it from that angle as well, using oats with the different protein and oil, and see how that affect the extractability of the protein. And we try to explore two methods which we are familiar with in terms of concentrating protein. One is wet extraction, and the other one is through dry fractionation through air classification, where we have pilot and the laboratory scale projects. So based on that, we have come up with the different protein ingredients with the varying protein from somewhere 60 to 92%. And we also look at the trying, even for beta glucan, varying from 2 to 18%. So depending on what the consumer may demand, we can produce a specific product for them. But what is more important to know is that when it comes, for example, to wet extraction, is that protein solubility and extractability is highly affected by genotype and environment as well as their interaction. So on top of that, we try to look at the, from protein quality, we want to see whether growing environment or genotypes affects the amino acid profile. As we could see here, from these three categories, you could tell that in as much all of them they're oats, but some of the amino acid tend to vary a little bit. Whether that can happen at a larger scale or not, that's what is interests us to actually find out. The other thing is with regard to versatility of oat ingredient. We know that it is applied in different products, and one of the concern we may have is when oat ingredient is subjected to high temperature or baking, where there's a potential of forming acrylamide. And one of the precursors to that is asparagine, which is the amino acid. And through Maillard reaction, it can form acrylamide, which is suspected to be a carcinogen or a foodborne toxin. So what we wanted to find out is whether this free amino acid asparagine is affected by genotype and environment, so that at the end of the day, we can relay that information to our breeders or agronomists to see what can be done to reduce the concentration of amino acid in our oats. So that's what, and our results showed that asparagine content is actually affected by genotype, environment, and the interaction, which brings us to the understanding that it can be addressed at the breeding program or at agronomic level as well. Then the other thing which we try to look at is the phenolic compounds, as well, more especially if we look at enanthramides. Why enanthramides? It's unique to oats. So that's why we think it has an advantage. And for it being like a phytoalexin, we know that its content varies a lot. So we did a G by E study to see how it affects the enanthromide concentration in oats, in Canadian oats. So for this study, we found that in Canadian oats, we had three major forms, enanthromide A, B, and C, and their concentration was highly affected by location. And the other part, when we look at enanthramides, the other question we had was how is it affected by, let's say, processing? And in this case, we're just giving you an example on terms of just a simple oatmeal preparation, where we know many people microwave it, or some people just boil it in a pot. Changing. Then the other area of research that we look at, and here we could see a good example mentioned. Normally for quality registration, it's mostly we look at the beta-glucan content, but we go deep into it in terms of quality, looking at the solubility and molecular weights. So we know that these two plus the concentration would have an impact on the viscosity. Why viscosity is important is that all the health benefits related to oats have been linked to oats' ability to form viscous alumino content. So here we could see that depending on how the oat is processed, the solubility and molecular weight is actually affected. Then lastly, we also look at the sensory properties of oats, looking at trying to look at the relationship between the instrumental analysis as well as the sensory characteristics from consumer panels. And we also look at the volatile components of oats in order to determine the oat flavor and aroma characterization. And so here you could see as well that if you plot them, they can actually, you can separate them, that at every process, the resulting flavor profile of our oat is actually affected. And finally, I will not go much into details here, is we are also working mostly on how to utilize starch, as we mentioned at the beginning, that we are working into protein concentration programs, which results into a lot of starch as a byproduct, and we don't want that one to go to waste. So we try to see if we can actually apply it into biofilms as well as noodles production. Thank you. Now I'll hand it over to Sijo.
Thank you, Lovemore and Nancy, for the introduction. I hope everyone can see my presentation.
We do see it. It's not in presentation mode.
Is it okay now?
Yes, it's perfect. Thank you.
Okay. Hello, everyone. And I'll be, as Nancy and Lovemore was talking about, I will be focusing more on the nutritional quality. Lovemore talked a lot about the end product quality. And we all know that oats are primarily consumed because of its health attributes, right? So the quality criteria around the nutritional qualities been evolved and been developed. So depending on the consumer demands and the health attributes, we are in a position to come up with, every day with a new attribute or new ways to screen or measure the nutritional attributes of oats. So with that in mind, I would like to introduce the interdisciplinary collaborators. We work with Dr. Nancy Ames, Lovemore, Thomas Nightingale with AFC, Dr. Jennifer Mitchell-Fetch, she's on the call, I think, Rebecca Petrek, and our industry partners, PepsiCo and Prairie Oat Growers Association. To start with, I will explain about our experimental platform we use for evaluating the nutritional quality of oats. It starts from the traditional analysis, compositional analysis, which involves the measurement of proteins, fiber, starch and lipids and micro and macronutrients chemically. And then we also have in vitro, static in vitro methods, which basically evaluate the effect of these compounds in a biochemical setting or in a physiological setting, how it responds is in a real life situation. So we use a cell culture, different types of cell lines, for example, endothelial and epithelial cell line like CACOs, and sometimes oocytes to study specific transporters of glucose, etc. We also have the next level is dynamic in vitro, or we call ex vivo, where we do have the stomach model, as you can see below, which is a dynamic system which simulate the human digestion process, where we will be able to measure like the digestibility, bioavailability, and the release of the nutrients during digestion. And the last stage, once you pick the right compounds or formulations, if you want to study the effects in humans actually or animals, we will go ahead and do this animal studies or acute human feeding studies to really validate our findings, which we find in any of these previous steps. So to further explain the in vitro experiments, we do have in vitro methods for glycemic response, kind of a modified English method. We also have enzyme inhibition assays to predict some metabolic activities. As I mentioned, we do work with cell culture to predict some of the physiological or biochemical outcomes of oat-derived compounds. To give you an example, one of the projects we are currently working on, Dr. Lovemore Malunga explained about it, the effect of oat enanthramides. So the compound we extracted from oats, we tried that on a specific cell culture. It's called caco cell lines derived from epithelial cells. We wanted to investigate whether enanthramides, which is unique to oats, has any effect on the glucose transport. We know that once you consume any grain or any food, the glucose is being released into the digestive tract, but ultimately it has to go to the bloodstream. It has to be transported as glucose to reach the blood levels, right? So we wanted to investigate whether any of these transporters are inhibited by oat-derived phenolics, and we did measure the activity of SGLT1 and GLUT2. Those are the two predominant glucose transporters in the epithelial barrier. So we found that actually in caco cells, glucose uptake was reduced by enanthromide B and C. So it can actually inhibit glucose at a very significant level. We also confirmed this using oocytes, where oocytes can be used to specifically express a specific transporter. So we did that with both the transporters, and what we found was enanthromide A, B, and C were able to inhibit the glucose transport in oocytes also. So that confirms that in vitro, the oat-derived phenolics can inhibit glucose release, which ultimately the outcome is whatever glucose is released, a certain portion of the glucose is only entering into the bloodstream. The rest will be eliminated. So it's a good thing for people with diabetes, and also for healthy people, those who really want to control the blood sugar levels or postprandial glycemic response. Coming to the next step, where we use the dynamic in vitro, which is the stomach model. Basically, it's a artificial stomach or small intestine, where you feed the formulations or the grain or the food preparations. It will be digested as it happens in human, with all the enzymes and with all the peristaltic movements and even with the temperature, pH, everything is automated. So the good thing is you will be able to measure the bioaccessibility, the amount of nutrients that is released, and also the bioavailability, the amount that could be potentially going into the bloodstream using the system. As Dr. Ames indicated, we were able to measure the protein and carbohydrate digestibility profiles. When we talk about protein and carbs, we always talk about the concentration. Sometimes it's not the concentration, it's the amount that is digestible and gets into the bloodstream. That's what most important, right? So the system will allow us to precisely pinpoint the amount of amino acid that's been released, the amount of glucose that can be reached into the bloodstream, so which will give a clear idea of different formulations and how it would affect your metabolic activities. It also helps us to precisely understand how the physiochemical properties of food acts in the GI tract. To give you some examples of the projects we are using, we are currently doing a bioaccessibility studies using a dynamic stomach model for using our system, and also examining the protein digestibility of the protein extract, which Dr. Lovemore Malunga showed using. And so we could do the in vitro PDCAS and all the amino acid score using this dynamic stomach model. Coming to the next level of experiments, we use animal models of metabolic syndrome, for example, diabetes, hypercholesteremia, or even hypertension nowadays, elucidating the biochemical mechanism of action. Because in vitro or even in humans, you won't be able to precisely tell the real biochemical mechanisms by which whether it's beta-glucan or enanthramides, mediate health effects. So, but in animals, you could harvest the tissues and we'll be able to look at the biochemical mechanisms. This is done in collaboration with Dr. Thomas from Agriculture and Agri-Food Canada. A couple of projects that actually running at the moment is we are looking at the health effects of oat protein, which Dr. Malunga showed in a high-fat, high-sugar fed rat model, because earlier studies have shown that it can actually, oat protein is capable of inhibiting an enzyme which is involved in cholesterol synthesis, which is totally different from how beta-glucan lowers cholesterol. So think about protein together with the oat beta-glucan would have two synergic effects to reduce the cholesterol, which will be another marketable attribute along with beta-glucan. We are also looking at the effect of oat proteins in spontaneously hypertensive rat, which is an animal model of human hypertension. So the preliminary results shows that there is a potential because of the peptides present in oats, which could be helpful in lowering blood pressure in humans. So this study will tell us whether there is any potential or could be another health claim for oats and barley derived beta-glucan or proteins. Then at the end, once we see any potential candidates or results, we go to the next stage, which is human feeding studies, where we developed test food. It's a very complicated process because when you formulate the diet for human studies, certain things has to be matched. For example, total dietary fiber. For glycemic studies, you need to match the total available carbohydrates. The protein has to be matched. So it's a very complicated process to come up with the test food formulations. And once we have that, we go with randomized control human feeding studies. Sometimes it's acute, it's a one-time meal. Sometimes it's habitual. For example, for looking at cholesterol lowering, a person has to consume a diet for at least 4 weeks. So this is done in collaboration with Chronic Disease Innovation Center in Winnipeg. A couple of projects to give you an example is effect of oat derived on postprandial glucose response in healthy adults. As I said earlier, we found this in vitro, and we wanted to see whether this oat enanthromide has the potential to reduce the glycemic response in humans as consumed as oatmeal. We are also recruiting participants for another study, which will evaluate the effect of oat beta-glucan in managing a healthy blood pressure. The recruitment is going on. As you can see below, these are the breakfast cookies made by Dr. Ames group, which contains 4 gram beta-glucan per serving. So this will be consumed for 4 weeks, and ambulatory blood pressure monitoring will be installed, and they will measure the blood pressure over 4 weeks of time, and to see if beta-glucan has an effect on reducing blood pressure, which actually originated from our animal study where we found some positive outcomes. Just to give you an example for how we translate our findings from in vitro to dynamic stomach and to humans, and how we connect that with the genotype and environment. Because what I want to emphasize is we are not going to buy a bag of oats and feeding people, because that won't give us the clear idea about how GBI affects the efficacy of bioactives. So for example, as you can see in the first box, we found the inhibition of intestinal glucose absorption by oat enanthromides. So then Dr. Lovemore Malunga and team went ahead and examined different genotypes and environmental variations to see if the phenolic acid and enanthromides are varied, and what extent, and would it be physiologically relevant when it consumed as a diet like an oatmeal instead of as a nutraceutical, because we are not talking about concentrated nutraceuticals here. We're talking about daily oat consumption, and can we achieve the physiological levels of bioactives. So we picked 2 varieties naturally, natural variations with high and low enanthromide containing oats, and we produced oat flakes out of it, and now making the oatmeal to feed for the clinical trial, which shows at the end a pilot study of evaluating oat phenolics in postprandial glucose response. So people will be recruited. By next week, we are starting the recruitment, and then we'll be fed with oatmeal containing naturally low and high levels of enanthromides. And we wanted to see, and making sure that the beta-glucan is leveled, the total available carbohydrate is level, but just difference in natural differences in enanthromide can have an effect on glycemic response. So we will update you the results later once it's out, but we are really hoping that enanthromide can inhibit the glycemic response as we see it in vitro studies. So thank you. So I'm happy to answer any questions if you have.
Thank you so much, everybody. That was really great. And it's really interesting now to see how oats are moving from, you know, more for the feed industry through sort of a minimally processed food item. And now it's being used in so many wonderful different ways, and that's going to continue. And I think that's really wonderful. So thanks so much for sharing all of that information with us. There's a lot of work ahead, but it's really exciting stuff. We do have a number of questions here. Actually, I will point out in the chat, I think everybody probably noticed the USDA ARS in Fargo, North Dakota is actually looking for an oat quality researcher. So if you're looking for a position to do that, please get in contact with them. The very first question here actually is, can the three of you send the presentations in PDF format after the webinars? I know that this is recorded, so people will be able to watch this again online, but I don't know, Nancy and the rest of you, are you willing to share?
Yeah, we'll put them together and share it. How do you want us to share it then?
It was suggested it be shared in PDF format.
Yeah, do you want it like sent, we'll send it to you then, or?
Yeah, if you send it to me, I can certainly, I can add it, put it in the Oat newsletter. Jim, I don't know if you want it for the Oat Global Speaking of Oats page, but there are
sure. And we can mail it out to anyone who's attended. Anyone who registered for this webinar will have your email address, and we can also email that out. Yeah. So thank you to the speakers.
Okay, or if there's just some people that want it, like probably everybody doesn't want to get this in their inbox. So I'm happy to give it to anybody that wants it. We're happy to give it. So.
Okay, so why don't we post it to the Oat News, and if you would like a personal copy, reach out to Nancy directly. Is that acceptable?
And I don't know if we're supposed to be like, I've been noticing the questions and I've been starting, okay, I'll start answering this, but I didn't know if that's what I'm supposed to do. So maybe you can just clarify that.
I'll read them. Yeah, so sometimes, yes, the panelists do answer the questions as we're proceeding through the rest of the presentations, but I'll go through all of the different questions here. So for example, yes, a number of them have been answered now, and I'll touch on those briefly in case you haven't all had a chance to look at those. So Dan Windrell was wondering if there are different enanthromide concentrations based on preparation method. Is this a solubility or measurement issue? And Lovemore actually answered this one and said, this has to do with extractability due to changes in food matrix. We are currently running in dynamic stomach model to see if that has an impact on bioaccessibility. So is there anything anyone would like to add to that answer? I guess not. Okay, so the next one, this was from an anonymous attendee. Is this quality research done anywhere else other than University of Manitoba? At this time, how do processors test for quality? Do they buy certain varieties that they know work for their end product? And again, Lovemore has answered this and saying, yes, most of the quality work is done at the Agriculture and Agri-Food Canada lab at the University of Manitoba. Depending on their intended end product, processors may have a preference on a particular bar.
I'll just add
to add there, sorry, meaning this, the data that we have presented here, and that's where I was pertaining to. Yeah.
Yeah, I don't think I'm not sure that's what the person's asking exactly. I'm not really clear, but it's almost like a great opening question to say that a lot of the, it depends there's lots of people working on oat quality, and possibly this is something, I did have a slide on that, and then I guess I didn't have time to show that, but even within Agriculture and Agri-Food Canada, if we just talk about our own organization, there's a lot of people that are working on oat quality across the country, and from many perspectives, plant breeders, people that are working on genomics, people that are working on quality with the breeders, people that are doing screening, people that are doing various types of research on the functionality of the protein. We have a number of research stations that are focused on this and focused on oats. And our own work in Manitoba, we're located at the Richardson Centre doing research work primarily around food quality and nutrition, but also working with all these different, other researchers, breeders, people that are agronomists, or people that are looking at processing. So, and in addition to us, of course, every company has their own, many companies have their own list of varieties that work best for their process, and they're constantly evolving that as well, finding possibly they're looking for new attributes because they've made new products, or possibly they're looking at augmenting some of their products, or now recognize the value of certain nutritional aspects, and they look at that. So how do people get this research done? Well, sometimes they are doing it in-house. Big companies have their own labs where they do this sort of thing in-house. Some people do research collaboratively with universities across the world, or with organizations such as our own, or USDA, or others. So there's lots of programs going on, and different people working on different things, and lots of collaboration. So I don't know if that answers what the person is asking, but certainly we're not the only people. We're just working on our only little part of this, and always willing to collaborate.
And that's what's so nice about this oat community, is that people generally are very willing to collaborate and share and learn from each other and so on, and move things ahead. So I think that's really great. Continuing on here, oh dear, Sasi Tajakovic, I think I may have butchered your name, I'm very sorry. Are there any studies or reports on asparagine content and resulting acrylamide in oat-containing products, oat milk, etc.? Are Canadian oats higher or lower in asparagine versus US oats? Any studies? And Nancy had mentioned that you have been measuring this. Lovemore, did you have something to add here?
Okay, so I think I would just mention that at the meantime, it's hard to tell. We haven't done like an inter-country variation, like a study to see whether US, Canada, Finland, Australia, whatever. It's a hard question to answer, but this one is only referring to what we studied within our Canadian oats varieties. And just an extension to what the other part of the milk, I would assume that asparagine will not be a problem with milk, considering that the processing itself is not a high heat. So that one will not be a problem at all. But here it's only referring to high heat. For example, as I mentioned, baking, that's where now it becomes a problem.
Right, yeah. Okay, thank you. There's a follow-up question here. Bitterness seems to be a problem when processing oats to oat beverages and other oat-based food products. Do we know the cause or causes and how to cope with them? Nancy, you had started to answer this one here. It was related to processing or rancidity. Would you like to add to that? You were suggesting.
Yeah, I'm trying to see where my answer is.
Under the answered tab.
Okay.
Q&A.
Okay. Well, what I was just saying is that what we've come across in terms of bitters, like the interesting thing about oat milk is, and I mentioned in the presentation that, you know, we're all scrambling to understand what the quality characteristics are of oat milk. And some people have a better handle, but, you know, it really depends on how you're making it. And there's so many different processes that are out there, so many different ways that people are pursuing this, that it makes a difference. And what we've noted so far, although it's certainly others might have other stuff to answer, maybe this is a good place for, you know, the rest of the group to add in. But what we've noticed is that we have seen some taste considerations, bitterness related to the way the oat is pre-processed, I suppose. And also, we've seen another case where it was really a rancidity issue, also related to the pre-processing. And I suppose, although we haven't looked at it, a good thing to look at is possibly the phenolics and whether they're contributing. But we haven't looked at that with respect, at least to oat milk. And I don't know if Sijo or Lovemore in particular has anything else to add about that potential, but I mean, we're just early on in this whole oat milk thing, but that's what we've noticed in terms of negative flavors, I suppose.
Yes, I think you are right, Nancy. So we are still trying to understand that issue. So at the meantime, whatever I can say will be more speculative. So.
Great, thank you. Switching gears a little bit for a minute, Chris Green, who's in England, is wondering, are the panel aware of any oat breeding programs with high amylose lines or varieties? Are any of those available? This might be somewhere where I don't know if breeders might want to weigh in or.
I'll just say that we've been screening lines and varieties for amylose, but we're not aware of any breeding programs that are focused on this for sure. Like I'm not aware, at least. And I'm sort of thinking that there could be some effort in Australia, but I'm not exactly sure. Somebody might be able to add to that. Maybe one of my colleagues or somebody else that's part of the whole thing here.
Thanks. And Nick Tinker would like to know if cadmium uptake is an issue for oats in Canada.
Again, that's one I don't know, Nick. I'm not sure of it. We don't measure cadmium regularly. We you know, not in oats. We haven't. Maybe Jennifer knows. Like I don't know about it, and it's not something we've looked at. Although we did look at it a lot in Durham, and yeah, interesting thought, an interesting advance in oat quality. That's one of the ones that needs to be pursued, you know, and something that somebody you know, that's here may be already pursuing or may be interested in pursuing. Great idea.
Well, Jennifer's on the call, so you know, Jennifer, if you have an answer, please pop it in the chat or even in the Q&A. Next, we have Anup Dakal. How much do you weigh the concentration of enanthromides in overall oat quality? Do you think it has a significant role in the quality of oat? Do you think the breeding program should start using this as one of the traits to be studied? Also, I'll add on the side, just for myself too, that enanthromide production, that's related to things such as crown rust resistance too, right?
Yeah, it's a phytoalexin produced in response to the stress. It could be the humidity, it could be the UV, could be the infections. So Yeah, with regards to its in overall oats quality, the studies are going on. I know many industries are looking at the concentration of enanthromides and its nutritional benefits. So probably could come in the future, but at the moment, it looks like a very potent molecule, which could be another beta-glucan, maybe in the future. You never know, right? So I don't know at the moment how much value it has on oat quality measurements, but definitely there is promising studies coming up.
Well, I would say that this is kind of why we're looking at it. It's an interesting way of Sijo just brought up about beta-glucans. So years ago, actually, when I even started my research work at Winnipeg, we weren't even measuring beta-glucan on a regular basis as part of a quality criteria. So things change. So why do we have a target for beta-glucan? Well, we have a target that is specific to the health claim. We need a certain amount of beta-glucan concentration to meet, so that the product can meet the health claim and, you know, give the people 3 grams of beta-glucan per day, and so that we can actually have a health benefit. So if we don't do this research, if we don't do it in advance, it doesn't make sense in my mind to say, breeders, okay, come on, I'd like to see you start screening for, the ratio of enanthromide C to be this much versus A and B, without us having a clear, possibly a health benefit that could be documented, that could be validated, or some other specific reason. So I think this is why we do some of these things. Some of them are partway there, and that's why I was mentioning it in the list of these are targets coming. Targets coming if we're not looking at this. We know very well that viscosity of beta-glucan is really the important thing in lowering cholesterol. But we also know that viscosity is not the important thing in lowering blood glucose levels. And so all of these factors have to be researched and documented and put into some sort of health benefit before we're necessarily going to be screening for them necessarily. We're coming close on a lot of these things. A lot more clinical evidence is coming forward. And this is why it's kind of important to do some of this stuff. But, when we go back to looking at flake quality, we don't measure that on a regular basis. And this is stuff we know, this is stuff we're very clear on, what flake quality is and how important it is, but we still don't measure that.
Thanks, Nancy. Just to follow up a bit on the cadmium question, Axel Diedrichson from the Gene Bank here in Canada has said that he'll be happy to screen the PGRC collection for cadmium uptake. So that's a good thing. Thank you, Axel. Also, Jennifer Mitchell-Fetch has said cadmium is something we were almost afraid to look at, but it probably should be checked into. And following on to the high amylose content question, Jennifer has also said, she's asking whether that would have any detrimental effects on processing. Do you know of any?
I can't, the effect of cadmium on processing.
Sorry, no, this is back on amylose, the high amylose.
Yeah. It definitely could have effects on processing, but it also could have, positive health effects, resistant starch and all that. That's one of the things that I believe Lovemore is looking at in his program now. So, but definitely something we would need to be looking at the processing angle too, because this is part of the, when we look at genotype, we also look at environment and we look at processing, G by E by P, and why all of those things together make a difference. But yeah, good point. It could have an effect and could be positive, could be negative, depending on the product.
Right, thanks. Just a very straightforward question from Sepper Naragi. What are the quality criteria for oat milk? Are there any right now? Anywhere? I don't know that there are in Canada, are there?
No. At the meantime, I think most
people are.
Sorry, go ahead, Nancy. Did you have something to add?
I was going to let Lovemore finish.
No, go ahead, Nancy.
I was just going to say that I know that there is, that some various companies are aware of that fiber certainly makes a difference, that, protein content, beta glucan is, makes a difference in depending on the way it's processed. So there's certainly not set criteria. And I guess part of the reason is probably about the very varying methods of processing, that it's not clear how everybody processes, that, that this could make a difference. But I do know that there are differences among varieties in some types of processing, that there are varietal differences in making oat milk. But I mean, it's, if other people have stuff to offer here, it would be great. Like if other people that are on the call have some thoughts, it'd be nice to hear them.
Sure, for sure. Okay, Tier Wiesner-Hanks has a question too. You mentioned the classic problem with the enanthromides. They can be so variable. In the G/E study, did you find that AVN levels could be predicted from environmental variables that are easily measured throughout the growing season? That is, could you look at weather station data and say, let's source from this area of Manitoba, it got poor rainfall at such and such developmental stage, which may have raised grain AVN content, that sort of thing. Are you perhaps thinking of heading in that direction? Will that work?
That does work. And I think that's exactly what we're doing. I think Lovemore and Sijo can talk more about that in a current trial that's they're doing.
Yeah. So actually it varies a lot, like environmental, or probably like Sijo mentioned in the presentation, like even for the trial, it could be same cultivar grown in one location, it was almost absent. Then in another one, it was the highest. That's where now we found like a control versus the other one. And one of the features which now we need to collect data, but what is appearing mostly it's about the rainfall pattern and the humidity. And we can see that in certain places like here in Manitoba, we tend to have higher enanthramides every year. Always in those locations, we tend to have higher enanthramides. Whereas in the western side, normally we see low enanthromide continuously. So it has to be, we suspect it has to do with the soil, rainfall pattern, and the temperature.
Thanks. And Nisa Saidi would like to know, and Sijo, you've just answered this here in the box, but I'll repeat it again. Is there any correlation between beta glucan and enanthromide content? And Sijo, you said you hadn't directly measured it. However, from the samples, low and high AV samples, we don't see any significant change in beta glucan levels, and we need to confirm this. Would you like to add anything to that?
Yeah, we haven't measured it like directly to see if there is any relationship. But from the samples, as I said, for the human feeding study, we needed to balance the beta glucan levels, right? Because we don't want to make sure the effect we are seeing is with the enanthromide. So when we measured that beta glucans, it was not that different. It was pretty close. So the enanthramides were very low and very high, but beta glucans levels were very consistent. But again, this is not specifically designed to look at that relationship, but probably in the future we could look at that. But from the preliminary findings, I don't see a relationship between beta glucan and enanthramides.
Okay, thank you. And I see there is another question over in the chat. Anup Dakal is asking, how do you measure the enanthromide? Do you have an internal protocol, or do you follow any standard protocol? And Sijo, you did answer that here, that you use a UPLC based protocol, and that, yeah, if anybody would like, you will share that method with them. Would you like to add anything to that?
Do people have our email addresses if they'd like to write us directly about something like that, a methodology? We're happy to share with avenanthromides, with avenanthromides or asparagine, and we have methods that we've developed in labs, so we're happy to share in most cases. So just let us know.
Could you maybe put your email addresses in the chat quickly? And then I see we are after 3 o'clock, we're starting to lose some people, but most people are still here. So perhaps you could each type your email address in quickly. See if those come up. And there are a couple of thank yous. There we go. There's Nancy's. Nancy.ames@agr.gc.ca. And then
I think that's only going to the panelists. I'll drop this.
Oh, you're right. It is
else.
only going to the panel. Okay, Jim is
that's okay.
redirecting. That's the trouble with having everything compartmentalized in these this webinar setup. Okay. Do you have the other two there too, Jim?
Yes, coming.
Yeah. Because yeah, the name of the game is communication. So, and sharing. And I don't just, okay, so there, everyone should be able to see the email addresses now. I don't see any more questions here or comments that should be noted, but I may have missed something. Is there something that I've missed?
The only thing I'd like to add, Charlene, well, I'd like to say thank you for inviting us. And again, we're willing to share whatever people are interested in. I'd also love to see some information or anecdotal information, even if people want to talk about oat milk, or if people have some things to say about it, or ideas and thoughts about what are important criteria other than what we're seeing varying from process. Like in many cases that we've worked on it, it'll be some kind of some kind of process that might be specific to some company that we can't share. But if people have general overall thoughts, it's great. It would be great to share that, because and again, it comes back to people. Often I get the question, is oat milk a trend, or is it here to stay? I mean, who knows? It's I might have said 2 years ago it was a trend. I mean, everybody's drinking it. And I really don't believe that plant-based protein, particularly oat-based protein, is a trend, because oat-based protein has a lot of advantages over, and we didn't have time to spend a lot of time talking about oat-based protein, but it's got a lot of advantages nutritionally, functionally, taste-wise. The taste of oat protein is good. And these are very important things. I don't think it's a trend. And of course, you use protein sometimes to make oat milk. So I don't know. I'd be curious. I wish we had a little doodle poll or something to say, is oat milk a trend? To hear from everybody on this call, it'd be nice to know.
Yeah, I think originally, one of the things with the Oatly brand was it had been developed for people with lactose intolerance, of course, but then now there are a whole lot of other reasons and different things going on. And I know that just from being on Twitter, at Oat Newsletter on Twitter, if you want to follow, yeah, there has been some controversy, different brands saying, well, these people do it this way and there's not very much nutrition, and ours has lots of nutrition, or these people are, there's some infighting, and there's some people saying things like, yeah, the whole plant protein thing is falling apart. And stocks are going down, all this type of things. I think right now there's a lot of turmoil, but I think overall, yeah, I think Nancy, you're right. I don't think it will be a trend. I think it will continue, but I think it is hitting a bit of a rough patch right now from what I've been reading. So hopefully that's sorted out soon. And again,
well,
even looking at those things though too, that might give people some ideas about how to reach their audiences, how to explain to people, you know, look, this drink has this oat protein, which has these properties. Like there might be some reason to look into, yeah, which are the better processes and how to, well, advertise, but make it known that maybe this is better than that and that sort of thing.
I think it's clear that the person who's left because they're off to join a glass of red wine, off to enjoy a glass of red wine, isn't going to drink oat milk. So.
Yeah, fair enough. So I don't see, I see a few more thank yous here. I don't, I did put the Oat newsletter email address in the chat, by the way. There are still quite a few people on here though. If anyone else would like to make a comment or ask a question, I guess you can go ahead. Otherwise, I suppose I would pass this over to Jim.
Thanks, Charlene. And just to set the record straight, I enjoy both oat milk and red wine, so they're not exclusive.
And I like dairy milk too, so.
I want to thank today's speakers for really lively talks, and of course, thank you to everyone who attended. A lot of great questions. It's wonderful to see the enthusiasm around this. Charlene, do you have any comment about December's Speaking of Oats, or is it still a work in progress?
Yes, I did have my little generic slide to put up. I don't think I'll bother at this point, but right now we don't have a confirmed speaker for December. We are talking to a couple of people that may or may not work out. I know in January, we're expected to have Pamela Zwer join us, and she's going to talk to us actually about the history of the International Oat Conference. So that will be something a little bit different and hopefully interesting for people. But yes, I'll let people know through the Oat newsletter and Twitter and so on what will happen in December. Also, Jim made this wonderful form for people to suggest speakers. If anyone who would like to give a talk, whatever, it would be wonderful if they could use the form. You can get to it either through the Oat newsletter site or through the Oat Global site as well. I don't know if you have a direct link you can put right there quickly.
I wasn't prepared for that, but I'll get it in just
well,
a
that's
second.
What I was thinking, right? So I might have it quickly too, actually. Let me just see if I can find it because it's in the newsletter. Let me see. Speaking of oats, I have a whole page there, speaker nomination form. All right.
Or do you have it?
I do. So that there, if that works, that should be the form to nominate more speakers. So please talk to people, you know, nominate yourself, tell us what you're up to, all that good stuff. It's really great to hear what people are doing and so on.
Absolutely. Well, thank you again for attending, and thank you to our wonderful speakers.
Yes, thank you very much. That was excellent.
Thank you.
Thanks so much.
Bye.
Bye, everybody.